introduce you to Meg Mindlin. And so Meg started out, from what I understand, growing up in Missouri, but then moved to California, where she went and did her undergrad at the University of California, Santa Cruz. And some point in there, I'm not exactly sure where, but at some point in there, she developed a very strong interest in one particular group of organisms that I also like quite a bit. Which, by the way, do you know what animal she is there? What's that? Actually, it's not an octopus. What's that? Vampire squid. Very good. Way to go. But that's not all there is to Meg. There's quite a bit more as well. In fact, one of the things that you, if you've been around this department much, but probably know, but maybe not, is that also Meg is quite an accomplished artist and has done a lot of artwork for a lot of different outlets as well. This is her website. I do encourage you to go check that out at megmindlin.com. And she does a lot of great stuff. This is one of the ones I especially appreciate because I can identify with this quite a bit. And again, it has an octopus in it. But just also to kind of plug there, she does have a merch store there too. So if you like her stuff, you can go buy stuff from there. So along with that, another interest of hers that has been very strong over the years is something that goes a lot really well with that with the art as well, and that's specifically she has a strong desire for science communication. It has a really strong interest in being able to communicate complicated scientific topics to a lay audience. So this is evidenced by her presence on social media. This is her on her TikTok. And so again, you can follow her there. She is the invertebrate. And so if you want to go, sorry, you seem to be embarrassed. But, I mean, to just demonstrate the level at which she is dedicated and really interested in science communication in particular, I think she was not in my lab but 1 week when she said, hey, I have to leave for a while. And that was particularly, she showed up at Rosario, was there for a very short amount of time, and then had to go down back to LA to participate in a Science Friday, an NPR Science Friday Cephalopod Week event that was happening down there. Which, by the way, if you don't know, is the other person there. This is Meg, 1 week into my lab, and then that's also another person from another academic university. That's Lloyd Trueblood right there as well, who is also in that. So, but not only that, she has been tapped to talk on Octopus in a lot of different venues. One of the most recent has been in the New York Times. She was interviewed, particularly in reference to this family that had accidentally, I would say, got a whole lot of octopus babies. And she was interviewed to comment on this, particularly because she was in contact with that family, but also because she's developing a presence known not only in academic circles, but very wide, of knowing a lot about octopuses. So here she is quoted in that. But that being said, I talked a lot about that. She is also, and I found this to be during her time in my lab, a very great budding scientist, and has spent a lot of time doing these kinds of things as well. And those are the kinds of things that she's going to talk to you about today. So with that, I'm going to give the time over to Meg and to tell you about, I'm going to knock this off myself, to tell you about all the really hard work she's been doing over the last couple of years. So here you go.
Now, quite literally, everyone and everything is affected by this. Worldwide, we are seeing increases in the number of natural disasters. We're seeing increase in the spread of disease, and globally, we're seeing a huge decrease in our biodiversity. Making this like not a really fun topic to talk about right now, so I apologize. But the one thing that people always ask me temperatures get too hot, octopuses have trouble successfully reproducing. So they're less likely to lay eggs, and of the eggs that do get laid, they are likely to not be fertilized. And unfortunately, this leads to a bunch of other problems as well. So right here on this graph, I have your x-axis as temperature and your y-axis as hatchling weight. And what we see is that of those babies that do get born in hotter and hotter temperatures, they become smaller and smaller. And a lot of that has to do with the fact that increased temperatures will increase the rate of development. So you have premature babies who then also have a much smaller egg yolk sac that they're attached to. And so this really impacts their survival in the wild, but also impairs critical physiological functions such as their vision, their speed, and their movement. And so in warmer temperatures, octopuses aren't doing too hot, with both reproduction, development, and survival heavily impacted. But what about ocean acidification? Because temperature isn't the only thing that is changing. Our oceans are becoming more and more acidic as well. And this is because all the carbon that we emit into the atmosphere is going to be absorbed by our oceans. She is by far our biggest carbon sink, sequestering 31% of our CO2 emissions. But in doing this, the pH of the ocean is going to be affected as well. And we're seeing this globally. Our oceans are becoming more and more acidic. And so while the effects of temperature on octopuses is crystal clear, acidification is a bit murky. And so when we're looking at the effects of high acidification in octopuses, we're seeing a few things. And one of those is going to be immune responses. And so on this graph right here, I have the x-axis as our partial pressure CO2. Week in acidification, we see that that rate under elevated conditions goes back down to normal. And at 5 weeks, it stays down. Now, this is interesting for a couple of reasons, but also in the context of another octopus, Moose octopus leoderma, otherwise known as the smooth skin octopus. Now, this is a deep sea species of octopus. They're typically found at about 300 meters, but currently we can find them in less than 10 meters off of the coast of Washington. And what's so interesting about the smooth-skinned octopus is that when we put them in the same conditions, they don't seem to experience any stress due to acidification at all. These same exact environmental parameters, they don't see a change in their routine metabolic rate, which is really interesting because despite the negative impact, there seems to be some resiliency to higher acidification. Now, this is most interesting in the context of all other fish, because fish do significantly worse in climate change scenarios, both under higher temperature and higher acidification. And one of the ways that we can see this is by also looking at their routine metabolic rate. So when they're put under very similar conditions as the last experiment, we see that their routine metabolic rate doesn't ever recover, even after 48 days, which is 2 weeks longer than the octopuses were exposed to, which is crazy considering that octopuses got their routine metabolic rate to go down after 1 week. So this is not great. It really sucks to be a fish right now. I feel terrible for them, but this isn't about fish. It's about octopuses. So back to the star of the show. Now, this resiliency is really interesting because despite octopuses' reproduction, development, and survival being negatively impacted by increased temperatures and ocean acidification, they seem to be doing okay. And that's true for squid and cuttlefish as well. And so one of the ways that we can look at this is through the different type of cephalopod populations. If we look at our demersal cephalopods, so those are the ones that live on or near the ocean floor, we can see that their population is increasing. When we look at our benthopelagic octopuses, so those are the ones that live above or near the seafloor, we can see that their populations are also increasing. And then again, we see this increasing trend in our pelagic cephalopods as well, the ones that never reach the ocean floor. And all three of these are confirmed by fisheries and survey data. So it's clear that octopuses seem to be doing okay, despite the negative impacts that we see. And so I'm really interested in this resiliency. However, there is another study that shows that if climate change scenarios continue, that cephalopod populations are going to see a sharp decline, especially in our warmer water varieties. Now, if you look at this graph right here, you can see currently where our cephalopod populations are. And the red indicates really high species richness. And you can see that's right around the equator. Now, if we look at predicted climate change scenarios, that same area has now turned blue, which indicates a real loss of species. Now, this model is only based on ecological niches, so abiotic factors, things like temperature and acidity, and it doesn't take into account biotic factors, like the fact that fish are doing much worse, and those are their primary competition. And so despite the fact that this is troubling, there are other interactions at play that contribute to a species' success other than just the environment they live in, though it does play a role. Those things are just a little bit harder to quantify and model. And so I want to focus on the resiliency because maybe I'm just a bit of an optimist, but to me that seems the most interesting. And specifically, that's why can octopuses acclimate to increased acidification over time? What is the molecular mechanism that is giving them a bit of a leg up on the competition? Now, I personally think it's RNA editing, which is just my opinion. I'm allowed to have it, but let me tell you why I think that is. So, if you're new here, this is the central dogma of biology, the sort of way of the land, if you will. And what it tells us is that our DNA is going to be transcribed into our RNA, which is then translated to our proteins. Now, I like to think of DNA as sort of the cookbook, and RNA is going to be all the recipes that are located within. And your proteins are going to be the finished meal that you've made. Now, me and all of y'all, the normies if you will, we primarily evolve through random mutation to our DNA. But octopuses have got a little bit something different going on, whereas they like to change the recipe book and edit their RNA instead. Now, octopuses aren't the only ones to edit their RNA. I'm putting them a little bit on a pedestal, I will admit. In fact, there's lots of other animals that edit their RNA. It's humans, mice, worms, plants. It's pretty widespread. But RNA editing is done a little bit differently between us and octopuses. So for example, in humans, editing is primarily done in the non-coding regions of their genome, places that don't really affect the proteins that are getting made. And this is mostly to prevent unwanted immune responses, so stopping a mistake before it occurs. But octopuses are editing those coding regions, the places that do change and affect their proteins. And now, this isn't entirely strange either. Other animals do have identified sites in coding regions that affect proteins. But what's the strange part, or rather interesting, is the number of sites that are occurring. So for example, in worms, we have approximately 10 identified protein coding changing sites. In mice, there's approximately 100. In humans, there's approximately 1,000. But in octopuses, there is approximately 100,000 of these protein coding changing sites, and with higher estimates at upwards of 600,000. So clearly, they have taken this idea and have ran with it, whereas we kind of think it's unnecessary. Now, to give you an idea of what's actually happening, editing is occurring primarily in their nervous system, so their brain, and specifically, a lot of the times, in their optic lobes, which are these two big portions of their brain that sit behind their eyes. Now, what will happen is, within their brain, the RNA is going to be edited by ADAR, which is going to slightly change the functionality of the proteins that it encodes, which leads to a lot of different versions of the same protein that have slightly different functions. And so with that, I'll give you a little bit more info on what ADAR is. It stands for adenosine deaminases acting on RNA, which gives you a little bit of an idea to its function. So ADAR, with combination of these double-stranded RNA binding motifs, will bind to the RNA over the nucleotide adenosine. And then what will happen is ADAR is going to chemically alter adenosine to inosine by cleaving a section off. Now, during translation, that inosine is going to be read as a guanosine because it's a biological mimic. Now, this is otherwise known as A to I RNA editing. And one nucleotide change doesn't seem like it would be a really big deal, but it can be. And that's because changing one nucleotide can lead to an entirely new amino acid, which is what our proteins are made up of. So for example, this AUA is going to code for isoleucine. If ADAR came around, bound to this adenosine, and chemically altered it into an inosine, it would be now read as guanosine during translation. So it was once isoleucine is now methionine, which can impact the proteins that are, the proteins that are encoded by these amino acids. Now, there's a few different ways that we have been able to quantify what is actually changing in RNA editing in octopuses. And that's primarily been by changing the temperature and seeing what's different as a result. And from that, we have a few proteins we've identified. But most of all, things are occurring like changing the rate of ions flowing in and out of a channel, changing the rate kinesin is walking across a microtubule, and also changing conformational changes within synaptogamine, which is a protein that's involved in synaptic transmission in their brain. Now, the commonality between all of these is that RNA editing is changing cells' ability to communicate to each other under increased temperature. But temperature's cool and all, but what about ocean acidification? Well, that's exactly what my thesis is trying to address. How does acidification impact A to I RNA editing, and what changes are being made as a result? Now, I can't really talk about my research without bringing up our previous graduate student, Ricky Wright, who did this original experiment and is what all of my results are built off of. And so what he did was he collected 12 octopuses from the Salish Sea. He exposed half of them to normal PCO2 conditions and half of them to elevated PCO2 conditions. An optic lobe sample was taken from all 12 and then sent off for high throughput sequencing. Then that sequencing data was put through a bioinformatics pipeline that was created by another graduate student, J.D. Sarawit, in order to analyze the results. And specifically, what he wanted to know was, how does editing change between the two treatments? And so that's specifically looking at the percentage of editing that is happening at specific sites. So let's say we have normal conditions, and this G right here is 20% edited under normal acidification conditions. Once we increase that CO2, that same site is now 80% edited. And so this is a significant differential difference between the normal conditions and the high conditions. And these are what I like to call acidification-responsive edits, or CO2 responsive. And so what he found was that the percent of editing actually decreases under acidification. And so this is similar to what we see in higher temperatures, that with increased temperature and with higher acidification, editing is suppressed. He also identified 12,000 editing sites that were differentially edited between treatments. So these are editing sites that change that encoding amino acid and show a significant difference in the percent that that site is edited between low and high PCO2. Now, I call these acidification-responsive edits, or CO2 responsive, like I said again, but just in case you need a reminder. Now, from those results, we received annotations of those editing sites. And those annotations made it seem like zinc fingers were overrepresented. Sorry about that. And so now I get to introduce the other star of the show, and why my thesis is titled what it is, which is zinc fingers. Now, zinc fingers are protein structures that are characterized by this binding of a zinc ion. They are otherwise known as genomic regulators, being able to attach to DNA and regulate the production of whatever protein is made from that particular sequence. And they do so by acting as a transcription factor. So this means that if a zinc finger is bound to a sequence, it can recruit or suppress enzymes that are involved in translation in order to make that protein. Now, the zinc fingers that I'm specifically looking at are known as C2H2 zinc fingers. And they're named this way because of their conserved cysteine and histidine pairs binding this zinc ion. And also, C2H2 zinc fingers are known as the second largest superfamily of proteins in the human genome. And so the really cool thing about them is that they can actually link together to bind longer DNA sequences, because one finger can only bind 3 base pairs. And so this actually distinguishes them from all other transcription factors that usually only recognize short and degenerate DNA sequences. Now, in order to put zinc fingers in the context of octopuses, one of the very interesting things that was found previously is that C2H2 zinc fingers are massively expanded upon in the octopus genome. And so just to give you an idea of how massive that is, we can look at our other animals, such as our lopatrophozoans. Now, our lopatrophozoans encompass all of the proteasomes within the clade spiralia. And so this is going to be our other mollusks, our annelids, our bryozoans, and our brachiopods. And so within that group, there is an identified 200 to 400 zinc fingers. And so next, we can look at our eutherian mammals, which is a subclass of mammals that have a placenta and reach an advanced stage of development before birth. So that's us, but it's also whales, elephants, shrews, and armadillos. Now, they have about 500 to 700 C2H2 zinc fingers. And that's considered like quite a lot because, reminder, those 500 to 700 zinc fingers make up the second largest superfamily of proteins within humans, with olfactory genes being the first, so your sense of smell. But where do octopuses fit in with all of this? Well, octopuses, and specifically Octopus bimaculotes, because that was the first octopus to have their genome sequenced, and it's what this data is based off of, they have 1,800 C2H2 zinc fingers, which is like quite a lot in comparison to everybody else, other mollusks included. And so, with that background of what zinc fingers are, why I was really hyped to see them in our data result, we're going to get into some of the three main goals of my thesis. Now, the first and foremost is I wanted to truly know how many zinc fingers were represented in the transcriptome, because those original annotations are based off of already pre-characterized proteins, and the vast majority of octopus proteins are not characterized. So it doesn't really give us a good say of how many zinc fingers there really are. So in order to find out how many there really are, I made some custom code that identifies C2H2 zinc fingers based on their amino acid sequence, because it's almost like a little formula that they follow pretty precisely, and just a few amino acids are changed in between. And so next, I want to know what those zinc fingers are binding to, because that's their whole purpose, to bind to proteins and regulate the production of them. But more specifically, I wanted to know that if those bound proteins could change at all with editing. Now, finally, if I could identify those bound targets of zinc fingers, I wanted to know how those proteins could impact their physiology and behavior. Now, this goal was a bit of a stretch for a master's thesis, I will admit. But I did find some things that I am really excited about, and I will talk to you more later on in the presentation. But first, let's talk about how many zinc fingers I actually found. So when I looked at the entire transcriptome, I found that zinc fingers take up less than 5%. Now specifically, that's like 1,500 zinc fingers, which is pretty comparable to the 1,800 that we saw in Octopus bimaculotes. So that was a good sign. And so slowly, I can start to narrow that window down to only just the edits that are responsive to acidification, meaning that they were significantly differentially edited between treatments. And when we do that, zinc fingers start to take up slightly more room at now 15%. And so at the smallest window, looking at only the top 50 edits that are the most CO2 responsive, zinc fingers now take up 30%. Which is quite a lot of zinc fingers, and it's really exciting, but is that really where the majority of our edits are occurring? Well, let's find out. So up here, I'll have two histograms placed back to back. On the x-axis, I have the percent of transcripts, and on the y-axis, I have number of acidification-responsive edits. And so when we look at just the transcripts that don't contain any zinc fingers, so these are genes that have no zinc finger domains occurring on them, we find that 95% of them don't have any edits at all, or at least under acidic conditions. And that there's only about 3% of edits that have one editing site, like 2% that have two, and almost no genes have more than 10 edits on them. But out of all the transcripts containing zinc fingers, we see a really big reduction in the number of sites that don't have any editing, from 95 to now like 78. And we also see an increase in the number of sites that do have editing. So now like one site or one edit is now at like 7%, 2 edits is at 5, and we see a big increase in the number of sites that have more than 10 edits on them. And additionally, we also find that zinc fingers edited in high acidification also see an increase in editing rates in comparison to normal conditions. So say you have a zinc finger under normal condition and it's edited at 30%. Under acidic conditions, that's now like 80 to 100%. So it's really clear that I was onto something, and that zinc fingers seem to be significantly targeted under acidification. And so both of these data sets can tell us a few things, just to recap. One of which being that zinc fingers are disproportionately more represented in transcripts with acidification-responsive editing sites. Secondly, genes that contain zinc finger domains are significantly more edited than those that don't contain any zinc finger domains at all in acidification-responsive sites. And so both of these together leads me to conclude that when RNA editing responds to acidification, zinc fingers are appearing to play a rather important role. But the real juicy stuff is what they're bound to. Because a zinc finger can only tell us so much. We need to know how they're interacting with other proteins and sequences. And so I found this information using a position weight matrix, which is software that's made specifically for this reason, to predict zinc fingers bound sequences. And what I found is that what the zinc fingers are bound to does indeed change with editing. And specifically, I found 28 zinc fingers that change what they're bound to with editing under acidification. And so this means that the functioning of those zinc fingers very much could be changing under acidic conditions. And so just as an example, so you can kind of see what I mean by this, in normal conditions, an unedited zinc finger is predicted to bind to protein A. And during acidic conditions, that zinc finger is now predicted to bind to a completely different protein. And both of these proteins could have two entirely different functions to them. And so this indicates a change in the need of the animal. Whatever's bound to protein B would supposedly be something that would allow the animal undergoing environmental stress to sort of adapt or acclimate better, hence the change. And so with that, we can get into what I actually found. And that is that within those 28 zinc fingers that had their bounding site change under acidic conditions, I actually got a lot of not very great predictions. And that's because, again, octopus proteins are for the most part uncharacterized. So anything that I bring up is not going to have really great matches to it because of that reason. And that brings me a lot of challenges in trying to identify the function of each of these changes. But I did get one site that I am rather excited about. And that is that when an unedited zinc finger in normal conditions was predicted to bound to a transferase enzyme. Now, these are enzymes that catalyze the transfer of atoms within a cell. Now, this one I don't really care about. It got a low confidence score. I just don't truly care about the unedited site. What I do care about is the edited site. Now, once that zinc finger is edited under acidic conditions, it's now predicted to bind to a protein that makes up something called the nuclear pore complex. And this one I received really great confidence scores for, so I'm pretty excited about. And so the nuclear pore complex is responsible for transporting material in and out of the nucleus. And so even if this one is not 100% correct, these are still two very different functions that can be enabled for very different things. Now, I find the nuclear pore complex interesting for a number of reasons. So I have it shown to the left right here. And like the name sounds, it is a pore that is in the envelope surrounding the nucleus in eukaryotic cells. So for anything to get out of the nucleus and into the cytoplasm, or vice versa, it has to go through this nuclear pore complex. And that's important to know because the nuclear pore is the middle ground between our RNA and our protein. In order for RNA to get translated into protein, the mRNA has to go in between this nuclear pore complex in order to get into the cytoplasm where translation is going to take place. And so the majority of this complex is made up by proteins called nuclear porins. And so that's kind of this whole thing. It's these little round guys, these little squiggly guys, these basket down here, and even these cytoplasmic filaments have little nuclear porins embedded within them. And so all of these different proteins work together to help transport cargo into and out of the nucleus. Now, the nuclear pore complex does more than that, though. It also helps regulate gene expression, not only by controlling the axis of transcription factors or the export of mRNAs in and out of the nucleus, but also by these nuclear porins just physically interacting with the nuclear genome. And so its role is both transport dependent and transport independent. And so what's so interesting to me about the nuclear pore complex in regards to my thesis is what other studies have found, which is that nuclear porins are some of the top regulation genes involved in adaptation to environmental stress. Now specifically, these nuclear porins were found to be one of the top regulation genes involved in adapting to high pH stress in Pacific white shrimp, but also in the two-toned pygmy squid in increased acidification. And so for me, that's the real icing on the cake, this study with the two-toned pygmy squid. Because specifically, when those squid were exposed to elevated PCO2, not only did they show that nuclear porins were to be genes regulating environmental stress, but that those genes were also correlated with the behavioral responses that they saw under acidic conditions, which was decreased speed, movement, and vision. So altogether, this explains or gives a possible regulatory pathway of how the nuclear pore complex is targeted by zinc fingers, whose mRNA is modified by acidification-responsive RNA editing. Now that was a mouthful, so let me break it down in little pictures for you, because that's at least how my brain works. So let's say we take an octopus who is experiencing increased acidification in its environment due to an increase in CO2 from human emissions. Now, that animal is going to undergo stress. And so we see that in an increased immune response and also a routine metabolic rate. And molecularly, the animal's body will respond by shifting what nucleotides get edited by ADAR. Now, this is the evidence that Ricky found. And so these changes in editing sites disproportionately affect zinc fingers, meaning that one nucleotide change more often occurred in the middle of a zinc finger domain on a gene than without. And this is the evidence that I found. Now, that change in zinc finger domains is going to alter what the zinc finger binds to, going from, say, that transferase enzyme to now a nuclear porin within the nuclear pore complex. Now, this is the evidence that I found within my 28 zinc fingers that had their bound sequence change with editing. Now, that nuclear porin is going to affect either what is coming in and out of the nucleus or directly interacting with nuclear genes by repressing or activating them. And this is the evidence that other scientists found when studying acidification in squid and shrimp. And what this means is that this very well could result in behavioral changes experienced by cephalopods under increased acidification, like speed, movement, and vision. And that's what we saw in squid in response to increased acidification. Now, altogether, this mechanism is based on the evidence that me and Ricky and JD and Kurt found in our lab, and also work that others have found in the field of ocean acidification. And so right now, this is just based on predictive and correlative evidence. So future experiments will be needed in order to validate it. Which I just gave you a lot of information all at once. So let's just take a step back and recap, going back to my goals and what I found. Now, I found a lot of zinc fingers, 1,500 of them to be exact, which is very similar to the 1,800 that was found in Bimax. I also found that zinc fingers are edited significantly more under acidic conditions than transcripts that don't contain any zinc fingers. And also, under acidification, there is an increase at editing sites with zinc fingers. Now, the second thing I found is that in increased acidification, the sequences zinc fingers could bind to could change with editing, and that one of those changes included the binding of the nuclear pore complex. And I've also identified a plausible mechanism for behavioral changes experienced under increased acidification. Altogether, this makes up the first study to identify C2H2 zinc fingers to be modified by RNA editing in response to environmental conditions that, that's the end of the sentence. And so if this mechanism is confirmed by future experiments, this would also be a novel mechanism by which RNA editing can modulate responses to environmental stress by altering the function of zinc fingers. Now that's all really exciting and really cool, but I started this whole thing off telling you how are octopuses going to be affected by climate change. And so the real question is, are they going to be okay? Well, my evidence doesn't really give us a concrete answer. It's hard to say. Octopuses and cephalopods as a whole are very much negatively impacted by the effects of climate change in their reproduction, their development, and their survival. But there are some factors that are giving them a bit of a leg up on the competition, one of which being their shown resiliency to environmental stress, and the other just being that they're doing better than fish. If fish die off, octopuses have more access to resources. And so while it does seem likely that cephalopod populations will decrease if our emissions don't decrease, there does seem to be a little sliver of hope that octopuses will survive in climate change conditions, and I think because of RNA editing, or at least it will play a very big role. Now, because of that, I don't think octopuses and cephalopods are going to go extinct altogether. But the best way to give octopuses, cephalopods, and other animals a fighting chance against climate change is to flex your constitutional right to vote, and voting not only in your federal elections, but your local and your state elections as well. And you want to vote with the ocean in mind, not only to help save the animals, but also for the success of our society as a whole. Because every degree of warming that we can stop is worth the fight and worth the effort. Now, thank you so much for coming here and listening to me. I want to do a special shout out to the previous graduate students who worked on this project, Ricky Wright and JD Sarawit. All the people who sent sequences to iTasker for me, that was a long haul. My collaborators who helped me with all the troubleshooting this project that surprisingly didn't drive me crazy. And I'm only as good of a scientist as the support system I have behind me. And mine is pretty great, which includes my partner Caesar, my mom, my dad, my grandma, and my 3 best friends, who are my biggest cheerleaders. And that's all. Yay. All right. Now, do we have any questions? Yeah, Ricky? Yeah, what is it?
So that edited zinc finger domain?
Yeah.
The big one?
Yeah.
How much do you know about the protein zinc?
It's part of that middle section of the nuclear pore complex, most likely. I think it was nub 155, which was also one of the nuclear porins that were identified in the other papers. It's, again, that middle portion, so those little balls in the middle. I don't have much other information besides just the fact that it came up in other studies as being one of the top regulation genes to environmental stress. But I don't really know how exactly it could aid in environmental stress, other than that shrimp one found a correlation between nuclear proteins and heat shock proteins. So that could be a possible mechanism for helping alleviate the animal undergoing high pH stress, but that's about all I know.
How about the zinc finger domain itself and the proteins?
If you could also repeat the question for people.
Oh, yeah. So the question is also, what about the zinc finger domain itself and the protein that it's a part of? So I did not look at where that domain binded to that particular gene, like where the domain is within that gene. I primarily looked at what those bound sequences we're binding to. And so I did that by getting the change in sequences and then aligning that back to the transcriptome. And I chose to focus on that side more so than the actual zinc finger side because I knew it was a zinc finger. I didn't really think I needed to know more about it other than that since I was primarily interested in those bound sites. So I don't got a good answer.
You don't know if there were any other edits in the zinc finger, like maybe in nearby?
There was a few. There was more than one edit in that zinc finger domain. There was 2. Yeah. I do know that much. Any other questions? Yes, Cals. Yeah. Yeah. I didn't see a pattern in like where those domains were located on genes. The pattern that we primarily saw was, the fact that those domains and edit sites were occurring more often in acidification than not. But in terms of a pattern on like those domains located within a gene, I didn't really investigate that any further. So no, I don't know. Yes?
There's one from online
Oh.
that says, what parts of the octopus's body are most affected by this editing? Any outward indicators? Could you spot one in RNA editing mode?
Oh, could you spot one in RNA editing mode? So the places that these are most often occurring is in the nervous system, so their brain. It happens in other parts, like for example, J.D. Sarawit originally did this experiment on gill tissue, and we saw a lot less edits. And so Ricky repeated it within the nervous system tissue, and we saw a lot more. In terms of outwardly responses, no. I mean, this is more so a thing that's happening without any sort of indication or knowledge from the octopus. It's not something that they're conscious of in the same way that you're not conscious of the little ALU repeats that your ADAR is editing in your body. So there doesn't seem to be any sort of indicator that RNA is happening within an animal in the same way that it's no indicator of us happening within us. Yeah. Is that everything?